Hi all, welcome back to Environmental Science 101. Today we're going to talk about margin of error and experimental bias. Okay, so this gets at the scientific method. So remember, we've been talking about the scientific method in the last few lectures, where we're talking about doing observations, making a question, developing a hypothesis, from that hypothesis designing an experiment, conducting that experiment, collecting the data, doing data analysis, and then from the data analysis drawing a conclusion. And then if the conclusion is support or not supporting your hypothesis, the final piece is publication. Well, along those lines of that scientific method, there are many situations where there might be error, or there might be room for bias. And so there are experiments and ways at which scientists go about to kind of prevent or minimize the amount of error and bias that's going in. Okay, so we know that it's really nearly impossible to account for every single variable in a scientific study, especially when these scientific studies are natural studies that are really common in environmental science, where you're collecting data that occurs in natural systems. In scientific studies that are more familiar in things like molecular biology, where you're doing things in a lab, or chemistry or physics, where you're doing things in closed systems, the accounting for single variable differences is a lot easier. Okay, but when you're talking about something that occurs in an environment or over a very long period of time, it's very difficult to deal with those. Okay, so that's why we have kind of an idea of a margin of error, or an estimate of how much error we're willing to accept. Okay, so we talked about this a little bit before when we were talking about statistics, and we'll keep talking about it. In biology and environmental science, in fields that typically study natural systems, ecology, evolution, often we have a confidence interval of 95%, or an alpha value of 0.05. So this means that basically we're accepting that there could be about 5% error in our design, in our measurement, and just that's the margin of error that we're willing to accept as kind of what I would consider the natural science. When we look at chemistry and physics, they're often a little bit more rigid, so maybe their margin of error, because they have more control over their experiments often, maybe their margin of error is 1% error, so their confidence is 99% with an alpha of 0.01. Okay, so that's where margin of error comes in. Again, it has to do with really how much error are you willing to accept, and we did an example of this in class. If you take a fair coin and you flip it 10 times, looking at heads to tails, you would expect it to be 50-50. But in many cases, you'll get something other than 50-50. You might get 7 heads, 5 tails, or 8 tails, even 2 heads, things like this. It doesn't necessarily mean that the coin's not fair. Remember, it's a chance, okay? And in this case, it's a 50-50 chance. However, if the coin is fair, the more times you flip it, okay, increasing your sample size. So instead of flipping it 10 times, if we flipped it 50 times, the closer we would get to that expected variable of half heads, half tails. Okay? And this follows suit with really all scientific studies. if you increase your sample size, if you increase how many organisms you're studying, how many data points you're collecting, how many days you collect your data, these different variables, then you decrease the margin of error. You reduce the effect of the margin of error. There's still error there. There's still going to be situations where, variables are unexpected. Okay? But the more samples you have, the less a single unexpected variable has an effect on the overall experiment. Okay? And so we can look at things like temperature. Temperature is a good example of this. If we look at temperature and we just go through, like, this is the temperature profile of New York, okay, from 1900, and we just look at the profile, we can definitely see that the temperature differences in the early 1900s, they were negative. They were less than expected, and now the anomalies are more than expected for the most part. Okay, so you can see that the temperature of New York City is actually on the rise. So one way to get at that margin of error is not by looking at actual temperatures, but by looking at how different are the temperatures from what you would expect. From that mean or that average, how much difference is there? Is it a negative difference, meaning that it's less than what you would expect, or is it a positive difference, meaning it's more than what you would expect? And in this case, it plots out fairly well that, as time has gone on, New York City's temperature is pretty much constantly above what you would expect as an average. Another way to look at this is when we're dealing with other data, like temperature data. Again, this is the temperature data for hottest places for Death Valley, and you can see that it was the hottest ever recorded was way out here in 1913 at 134 degrees Fahrenheit. Okay. Again, you can see as you look across the data, you can look from the 1920s all the way to 2020, and you can see that the temperature is on the rise, that you can see this kind of drifting upwards, and instead of looking at a snapshot on time and just concentrating on these outliers, like you can say, all right, well, the hottest recorded temperature ever was 1913, 134 degrees. Okay, so that must mean that the climate's not changing in Death Valley. Well, if you look at, instead of like a single point, but if you look at all the points together, again, this is getting at sample size, collect all the points for a long period of time, you can see that it's drifting upwards. You can also do things by looking at, well, how many days above 130 has there been, okay, or can we look at how many days above 120 has there been? And by examining the data in that way, you're compiling the data to not account or not have one single date or one single point create or have so much worth to it. Okay, so instead you're looking at averages or means, which would show that the mean temperature in this region is actually higher now in the last 20 years than it was in between the years of 1900 and 1920. Okay. All right. So other ways to combat biases. So again, one way to do it is collect more data. Okay, have higher sample sizes. That means the outliers will have less pull on your data set. Okay. Another way is to actually design the experiment to control for biases. Okay, so if we know there's going to be a bias in the experimental design, then pick a way to control for that. So, for example, in environmental science, we're often interested in, maybe we're interested in one river system, and we might be interested in the effects of logging on that one river system. Well, we can have a natural bias. We can go to regions where we're like, all right, well, they've logged right next to the river here, so we're going to set sample sites here, 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 and that's all we're going to sample. Well, but that's a bias. Okay, we're interested in the effects of logging on the entire system, not just a point, not just where logging might have occurred along the stream, but the entire river. So often when we're interested in a question like that, we'll have random points generated and random experiment points. Okay, so it's just random where those points lie. Some are going to lie next to the logging site, some are going to lie above it, some are going to lie below it, but it'll be randomized, and that removes the actual bias, the human bias put forth. We do this a lot, controlling for biases, a lot in like drug treatments and molecular data or molecular experiments, pharmaceutical experiments, medical experiments and things like that, because there are a lot of biases when it comes to humans as the subject. Okay, so if you know as an individual that you're receiving a placebo, so if someone told you, hey, you're either getting this medicine that controls your blood pressure or you're getting a sugar pill, and then they told you right away you're getting a sugar pill, then you might act in a different way. You might bias the research. You might either exercise more or a lot less. But you might adjust your behavior knowing what you received. So instead, what often we do is we do what's called a blind experiment. And that's where the subjects don't know if they are receiving a placebo or an experimental drug. Or they don't know whether they're in the control group or the experimental group. Okay? And even better yet is to do a double-blind experiment. And that's where the scientists don't know either. Okay? So the scientists don't know what group received the sugar pill and what group received the pharmaceutical drug. And so both parties, the subjects are blind to the study, and the scientists that are collecting the data are blind to the experiment or the study. That eliminates a lot of bias. Because, as a scientist, if you work for a corporation and you've worked to develop a drug, you would like to see that drug be successful. And so you may have some underlying bias that you want to see the drug win. You want to see it help people. And so if you know which group might be getting that drug, then you might interact with the data in a different way, okay, a biased way. Okay. All right, and that really brings me to scientific fraud. And sometimes scientific fraud is the result of biases. Sometimes it's the result of pseudoscience, but often because in true scientific method form, because at the very end when you go and you publish and that publication is reviewed by other scientists, normally what happens is we catch any scientific fraud or any biases that might come forth. Now, that doesn't mean that everything is caught, but real blatant studies that are have high biases or are the result of pseudoscience are often caught, and then that material is rejected. Okay, one that is a classic example of this was a paper done by Andrew Wakefield, and many other authors or et al. Andrew Wakefield's paper was really the first paper that was, or first research study or pseudoscience that linked measles, mumps, rubella, or the MMR vaccination to autism, okay? And other things like colitis and other gastrointestinal diseases, okay, or disorders. And his research, when it was published, got a lot of attention, and a lot of individuals were really concerned that by getting vaccines that they were increasing the likelihood of their children getting autism, increasing the likelihood of their children getting colitis, and all these things. However, after scientists got a hold of the paper, reviewed it, so this actually made it through peer review, okay? But scientists read the paper, and then the other nice thing about the scientific method is at any time, okay, people can ask you for the original data. At any time, people can ask you for, hey, I need your data analysis. I need to see where you get these numbers from. I need to see more of your experimental design. It's not clear how you did this. And that is exactly what occurred with the Wakefield paper. Scientists got a hold of it, found out that the science was pseudoscience at best, that the experimental design was poor, that many of the children who were picked for the study were biasly picked, in the fact that prior knowledge about the children was already known, some of which we already knew they had colitis, they had autism, and they had received the measles, mumps, rubella shot, and so it was not a controlled study. And the Lancet, which is a journal, a medical journal, retracted the paper. Okay? Other things occurred in the Wakefield process, but again, the purpose of the scientific method and that last piece of publication is the peer review. Okay? You got to have scientists read your material, review it, and see if there are biases or flaws or fraudulent results or pseudoscience going on. And unfortunately, this process means that a lot of science goes long periods of time before it's published, because the process of peer review takes a long period of time. So that increases cost. But when a paper like this goes through, it created a huge wave of skepticism on vaccination, and that wave still continues today, which increases the amount of mistrust in science, especially because the paper was originally published. Okay, so it made through the first review, or first few reviews. Normally you have 3 reviewers, and then publication. And it wasn't until later, after publication, that it was retracted and it was removed. Okay, Wakefield himself, like I just said, it was the purpose of the paper was to document this link between vaccine and autism. Wakefield, due to this paper and other not so medical doctor patient ethics, I guess, Wakefield lost his medical license in Europe, and because of the work that was done here, and how he went about collecting data from these children, and really the fraudulent results that came out of this. Okay, so that's a fairly good example of a scientific fraud that went through, got published, created kind of an outcry, and that outcry still currently today. There are many people that will not vaccinate their children because they believe the link that was put forth between MMR vaccines and autism in children, even though it's been retracted, refuted, and there's no scientific evidence that this is true. Okay. Another one that happened fairly recently has to do with what we call island or heat islands, okay, urban heat islands, and Wei-Chung Wang and his colleagues had a study that they put forth that looked at temperatures of urban environments, which they were calling these urban islands, and how that these urban islands, their temperatures are increasing over a period of time, and that these urban regions are actually what's causing much of the global climate change, much of the heating of the planet, are these large urban areas where they become this heat source, and because of the amount of blacktop, the amount of concrete and metal and things like that that are on the landscape, they absorb the heat and they release the heat, and they maintain warmer regions than rural environments. And really, the paper was a great paper, but the problem with it was that when reviewers looked back at it, they were looking for these sites. Okay? They had 42 urban sites and rural sites, but some of the sites, the stations were not available. So researchers could not actually see the temperature stations. Okay, so there were like 6 or so stations that could not be located. And when the authors were contacted about it, the authors couldn't locate those stations. And in fact, the authors later sent out a kind of a rebuttal to the review of it and said, well, that's because we got that data from other individuals. And so the fact that you couldn't reliably track down the information meant that this paper, at least portions of this paper, was did not follow the scientific method, because it was not repeatable. You couldn't find and repeat that data. You couldn't pull the data from all these stations because some of them didn't exist, or no one could locate where the station was, because it was basically just data that was given to the authors, and they didn't find out where the source got that information from. Okay, so just ways at which we try to prevent biases is by blind experiments, double-blind experiments, increasing sample size, but then the peer-reviewed process really gets at hopefully limiting the amount of biases, flawed, outright fraudulent results that occur in science, because people are people, and will cheat the system when they can. Okay, next time we're going to talk about energy and the environment.